I will talk about a user case of using ChemAxon and actually other software products as well. Uh, so the main question was that does a 3D shape-based method when comparing molecular structures differ from a descriptor-based method in its ability to predict biological activity? I guess most of you are familiar with what these methods are, but I will go into it as well. And the first answer is it's always database dependent. So we can only answer this question for a certain database. And the database we were looking at was the DTP's NCI 60 panel, which is a collection of 60 human cancerous cell lines. And it was originally developed to identify potential anti-cancer agents. And I think one of its main strengths lies in the fact that so many data is available from this set. More than 100,000 chemical compounds and natural products have been screened, and many measurements have been done on these compounds. In this talk, I will concentrate on the drug toxicity. If you want to think about it in mathematical terms, so there are the 60 cell lines, and then there are quite a lot of compounds for which the toxicity data is publicly available. So we will have a big matrix of toxicity values. And now I will talk about what was in the title. So what similarities we have defined. We can define a biological activity similarity. It has been defined in many different ways. So you can think of it as a binding capacity to a known target, or the gene expression changes as a result of drug treatment. There are other options as well. But what we had here was the drug toxicity across several cell lines. This is a drug toxicity measurement. On the y-axis, you can see the percentage of cell growth, and on the x-axis, you can see the concentration of the drug, and the log GI50 value can be defined as the drug concentration where half of the cells survive. And this data is available for the NCI 60 cells. I have just plotted some of these data for you. Here, if you look at the cell line index, as you remember, there are 60 different cell lines. And on the y-axis, this is the PGI50 value. And you can see there would be cell lines for which this PGI50 value is quite heterogeneous, and there would be others where it's really homogeneous. How can you compare these with each other? One option is to use the Pearson correlation. If you plot the PGI50 values of one of the compound and the other compound, you can calculate the correlation among them. For this, we have only kept those agents for which the PGI50 values were heterogeneous enough. Otherwise, the Pearson correlation would be falsely considered as too low, whereas using common sense, if you look at these two results, they are actually similar to each other. So that was the, and we can also look at structural-based similarities. Just really going through this because this is, I guess you are all very familiar with it. The molecular descriptor-based similarity, the molecular structures are converted into bit strings, and then these bit strings are compared to each other, and I've put on two examples here, the chemical fingerprint and the extended chemical fingerprint. And once the hashing algorithm has been done, and there is a fingerprint, and these fingerprints can be compared to each other, again using several different metrics. In our work, we have used the Tanimoto metric, which is defined here. This is conceptually, sorry, a completely different similarity, the 3D shape-based similarity, where you actually look at how well the shapes of the molecules overlap. There are advantages of this. So you are really looking at the molecule shapes itself. But on the other hand, in most cases, the active conformation is unknown. So we thought it's useful to use it, but we can't be sure whether it would be better or not than the molecular descriptor-based similarities. What is important for these metrics, it actually takes much longer computational time to compute them, because again there are different methods for this. The ChemAxon Screen 3D uses a pairwise comparison of the molecules and then just tweaking the molecules until the best overlap is achieved. And how we use this on our data set. So there was a data set preparation. Nearly 50,000 compounds were available with both toxicity data and structural information. But for some of them, it was only known for a few cell lines, which were omitted. And then there was this, we wanted the GI50 values to be variable enough. And then again, this is actually a quite heterogeneous database, so there were some corrupt structures which we had to remove. And in the end, we worked with approximately 5,000 different structures, what I will show you now. So back to the original question, we are interested how well structural similarity may predict the biological activity similarity, which is a general question, of course, and many works have been done around this question. And one way to assess this is look at the positive predictive value. It can be defined as the fraction of the number of molecules that are both structurally and biologically similar to each other, divided by the number of molecules which are structurally similar to each other. If you look at one of the structural similarity metrics and you plot a histogram of the molecule pairs, then there would be a certain number of molecule pairs. Here it's right to this line, so denoted by green, which display structural similarity higher than a given threshold. For these molecule pairs, the biological activity similarity can be plotted as well. So I have only plotted it for the green ones. And again, we obtain the positive predicted value as the fraction of those molecule pairs which are also biologically similar. It's right to this line, divided by all of these molecules which were structurally similar. So does it make sense? But of course, this positive predicted value would be very different at different threshold levels. So if we pick different structural thresholds, then you can see that the fraction of this and this whole area would be very different. And here I have plotted the positive predicted value along between 0 and 1. So that's the limit for the similarity values. And you can see that it changes very, very much. Why is it important? If you think of it as a virtual screening settings, then this would tell the fraction. So we would get a certain number of molecules as hit molecules, and this would tell the fraction of those molecules from the hit molecules which are actually biologically display what we wanted. There's another important metric, the sensitivity. this is defined as the number of molecules which are simultaneously both structurally similar and biologically similar to each other, divided by the number of molecule pairs which show biological activity similarity. So again, if you think of it, now on the first histogram you can see the biological activity similarity, so we can define those which are similar biologically, and then again just plotting the structural similarity of these molecule pairs, we can see what percentage of them will be also structurally similar. Again, just as before, it changes a lot if either the structural similarity thresholds or the biological activity similarity thresholds are changed, and it will display a completely opposite trend. Why is it important? Again, in a virtual screening setting, this would tell us that if we had a certain number of molecules which displayed the desired biological activity as compared to a lead, then what is the fraction that we can actually obtain from the screen. And obviously, our aim would be to maximize them simultaneously. Well, we can't do that because as you can see, the trends are opposite. If we change either the structural similarity or the biological activity similarity thresholds, while the positive predicted value will rise, then the sensitivity will decrease, and vice versa. So for the rest of this study, we selected thresholds which were kind of in the middle line, so reasonably high positive predicted value and sensitivity. And back to the original question again. So how well can these different methods predict the biological activity similarity? If we look at the shape of these curves, so now I've shown it for one randomly selected 3D shape-based method and one randomly selected molecular descriptor-based method. And what you can see that the positive predictive values and the sensitivities at these selected threshold values look very, very different. So can we say that based on these plots that their ability is actually different? It's a good question. I would say not yet, because if you look at, again, the distribution of the pairwise similarities, they are different. So they both show normal distributions, but their mean and standard deviation are slightly different. So if we change the previous plot to display structural similarity percentiles instead of just threshold chosen for one of the metrics, then you can see that the outline of these curves are quite similar to each other. And so we can say that the different similarity metrics are comparable in their ability to predict the biological similarity. And then the next question comes then, does it mean that they will actually select the same molecule pairs to be similar to each other? Well, they're not. So if we plot, again, randomly selected molecular descriptor-based, 3D shape-based similarities and biological activity similarities, then you can see that there will be molecule pairs highlighted in the black ones. Sorry, this is a typo here, which we can say are coherent. So they show a biological activity similarity, and they show similarity based on both structural metrics. So yes, we are happy with that. But what about these other octants in this plot? Interestingly, there will be molecule pairs that are similar based on their molecular descriptor-based metrics, but dissimilar based on their 3D shapes. So why can we obtain such molecule pairs? When we looked at it, we found out that mostly it were structures where there was a relatively large substructure, maybe occurring twice in a molecule. So that could warrant further investigation, because it might show the important active parts of the molecule, but we didn't look into it in more detail. And then this blue set of symbols display the molecule pairs which are structurally dissimilar based on both metrics, despite their biological activity similarities. Well, it's not a big surprise. We know that many of such molecules exist, which are structurally similar but display similar biological activities. They could be molecules which have targets in the same biological pathway. And then they could be, again, ligands of protein pump, or so there are many explanations for this. So it's we were not surprised to find this set of molecules. But what we thought was very interesting, that there are also molecule pairs for which the 3D shape-based similarities were high, and the biological activity similarities were also high, despite their low molecular descriptor-based similarities. And we thought that this could be putative scaffold hopping pairs. What is scaffold hopping? So it's a concept of modifying the central structure of a molecule while retaining its original potency. This is widely used in drug discovery, and we thought that it's kind of might already be there for these molecules, because they display a similar shape. We know that their biological activity is similar, but their molecular descriptor-based similarity is low, which could just mean that there is a central change in their scaffold. And we have looked at potential scaffold hopping analogues of FDA-approved drugs using this method, just selecting an FDA-approved drug, and then around this selecting other molecules which were similar in their shape and their biological activity, but dissimilar in their molecular descriptor-based similarity. And for which I will show you the results now is mitoxantron. It's an FDA-approved drug who's poisoning topoisomerase. And also, a crystal binding study is available for binding when mitoxantron is bound to topoisomerase 2. So we will be able to do in silico docking validation. And I've just plotted here the different chemotypes which we have found using this method. So you can see that there are typical scaffold hopping differences, either heterocycle change here or only a heteroatom change here, and so on. And for some of the molecules which are circled or in a red rectangle, because they were all DTP molecules, we could just get the actual molecules from the DTP. Okay, there are some more chemotypes again. So, for example, here you can see ring closure, which is again one type of scaffold hopping. And when we look at the docking scores, so what you can see here, these are the docking scores for the putative scaffold hopping molecules. Docking scores can be interpreted as when compared to docking scores of other molecules. So usual strategy is to use decoy molecules which have similar physicochemical properties, and the gray one, which is here, are decoy molecules obtained from the Jude-E database, which was specifically designed to obtain decoy molecules. And then we have created other set of decoys. So the red ones are molecules which are similar to metoxantron in their 3D shape, but not in their biological activity, whereas the blue ones are those that are similar to metoxantron in their biological activity, but not their 3D shape. So we wanted to test whether we need these two similarities jointly to obtain potential scaffold hopping molecules. And the results point to that. That's possible, yes, because the docking score, I mean, here it's enough that the smaller score is the better. The docking scores are in general smallest for the possible candidates. We have also looked at an in vitro assay. For topoisomerase 2, there is this specific assay when there are catenated DNA minicircles. Then this is what the enzyme does. It will decatenate them. And if the topoisomerase enzyme works, then from original catenated DNAs, we would end up with decontaminated DNAs, both nigged open circular ones and relaxed ones. So if we want to use at a gel photo, then when topoisomerase works, we kind of expect this kind of pattern. And this is the pattern we have obtained. So here what you can see is the topoisomerase enzyme. So without the topoisomerase, nothing happens. When it's added, then all of the DNA minicircles will be decatenated. This is a known topoisomerase poison. So if it's used, there's a lot less decatenation. And these are some of the potential scaffold hopping molecules which were highlighted by red previously. And actually they worked in similarly to the known topoisomerase inhibitor. So again, this also pointed that these molecules are really topoisomerase inhibitors, which is the main mechanism of action of metoxantron. So we could say that the obtained molecules would be really scaffold hopping variants of metoxantron. So in summary, it looks like that the abilities of the different structural metrics are similar among these DTP agents when predicting biological activity. And for us, those molecule pairs that displayed high biological activity and 3D shape-based similarity were of special interest. So we further looked at them and found collected these putative scaffold hopping analogues around FDA-approved drugs. And specifically for metoxantron, the in silico docking and in vitro decatenation assays point to that direction, that the mechanism of action is similar of metoxantron and the scaffold hopping analogues. And so we all work together in this project, and I was given a server from Gábor to run the calculations on, and of course I'm happy for the encouragement of my group members. So thank you for your attention.
If taking practical result out of it, if let's say I have thousands of molecules and compare result of similarity with standard similarity method with 3D shape similarity, can you estimate how many molecules will be the same and how many additional similar molecule I can find with 3D similarity?
you can estimate it if you have a main molecule to which the other molecules are compared to. So in the case of mitoxantron, it was in around several 10 of molecules. So the predictions of the two metrics are really similar in most cases. Only quite a low percentage will differ.
So conclusion is that both method will found the same molecules, but if I use 3D similarity, there will be some small number additional molecules that actually similar.
Mostly they will provide similar results, but there will be molecules which are only similar based on their molecular descriptor metrics and some which are only on their 3D shape. So it's always like that. So you use different methods, you will have differences.
If you have 2D similarity, the molecule is always very well defined. But then imagine 3D shape of the molecule. Like most of these molecules have lots of atoms, so I can imagine like several quasi degenerate minima, which can differ by like turning some group or part of the molecules. So my question is whether you have always considered just a single 3D shape for every molecule, and also whether on some occasions it would be better to use like several possible structures for the molecule to calculate them like among themselves.
So these are all very good questions. This is the general disadvantage of the 3D shape-based method. So when two molecules found to be similar, this is based on a certain conformation. Many different conformations are tested. So the molecules are tweaked and rotated around their flexible bonds and so on. And we have always accepted the highest similarity. However, we don't know whether that would be the active conformation or not.